to have a chance to be here today to share our early data of focused ultrasound neuromodulation for drug resistant epilepsy. So this is my outline today, and in the beginning, I would like to make a brief introduction of my site. And it's the largest epilepsy center in Taiwan, and fulfill the criteria of level 4 in National Epilepsy Center in the United States. And we have more than 200 cases admitted for pre-surgical evaluation every year, and among them, more than 80 cases were submitted to surgical intervention annually. And we are always keeping up with the time in seeking solutions for drug-resistant epilepsy. So in 2017, this is the first time I contact with Arthur, the staff of NaviFast, and then after discussion, we have some idea. So we applied a proposal to our Taiwan FDA and got approval to carry out the first pilot study of focused ultrasound for drug-resistant epilepsy. And in 2021, and we got another approval for the pilot single-blind randomized crossover studies, and which has been completed this year. And in 2022, we got the funding from our NHRI. They allowed us to investigate the brain connectivity modulated by focused ultrasound. We're using a tool called cortical evoked potential. And also, this year, our site, we got two approvals. One is using the NaviFast again to study the efficacy of neuromodulation effect of the low-intensity focused ultrasound on patients with drug-resistant epilepsy. And also we use the MRI-guided focused ultrasound to lesion the brain substrate to cure, to improve seizure control for patients. This is our first pilot study. I will make a brief introduction here again. And patients who have finished their intracranial EEG for drug-resistant epilepsy with the conclusion of their epileptic network were enrolled. And before the treatments, there will be a simulation planned. On the day of the treatment, we share the hair and register the coordinates by the Medtronic S7 navigation system. The operator will hold the transducer and adjust it to fit the pre-planned trajectories. And this is our protocol, the setting we use in the first pilot trial. 10 minutes is a little bit longer, but acceptable. And we analyze the intracranial EEG 10 minutes before sonication, during sonication, and 10 minutes after sonication. And in the target electrode here, we can see the red line here, the lower frequency power reduced after the treatment compared to before the treatment, while in the non-target region, there was no significant change. And in the second clinical trial, we just finished this February. We used a single-blind randomized crossover study. Why crossover? Because we expect there will be enrollment difficulty if you use a sham control study. So I tried to use this crossover, and all the participants will receive the treatments eventually. And this is the inclusion criteria and exclusion criteria. The target of the treatment is determined by the PI after a comprehensive pre-surgical evaluation. And this is our study procedures. On the day of the treatment, the first treatment, the patient was randomized to floss treatment or sham treatment and switched to another arm 4 weeks later. And the setting is much similar to our previous pilot study, but the sonication duration was divided to 3 5-minute sonication separated by 2 5 minutes resting. And 12 cases were enrolled in this trial. This table shows the demographic and seizure-related characteristic of these 12 cases. This is the result. Now, very good for first focus ultrasound. We observed the 4 weeks seizure counts, and there seems no difference between the focus ultrasound and the sham on 4 weeks after the treatment. And especially for the sham group, there seems a reduction of seizure in the sham. How do I believe that everybody will ask about this. I will say my explanation, part of my explanation is that the placebo effect and carryover effect in the crossover trial can be one of the reasons. So until now, the study should be over, and I was a little bit disappointed. But one day, the case 7 came into my office and said she was so excited. She has been no seizure in past 2 months after the treatment, which has never occurred in her past 2 years. So I think maybe I stopped too early. Then I extend the observation period. This graph, seizure counts over time, is pretty messy. But if you look closer, seems something here. 1 month end of studies. There's a trend over here. So make it clear. I normalize the seizure count to the seizure frequency % change from the baseline and align the treatment to the focus ultrasound. This is the result line. Then it is much clearer now. You can see most of the patients 2 months after the sonication, it seems the trend of seizure reduction here. So I submitted all the group into statistic analysis. Not very strong, but you can see that all 11 cases, the seizure reduced over time, and there is a significant statistic results, especially 4 months after the treatment, the seizure reduction is significant compared to baseline. Among the 11 cases, 7 cases have target in the medial temporal region, hippocampus, and another 4 in the non-medial temporal regions. As the line you can see here, it seems that the medial temporal group has better response compared to the non-medial temporal region. We also collected the self-administered anxiety, depression, and quality of life scale. Everything was non-significant except for the sham group. There was an increased anxiety score 4 weeks after the treatment. And for the adverse effect, 2 cases reported headache after treatment, which lasted for a few weeks and subsided. And 2 cases reported a sensation or headache during the treatment. And 1 case encountered technical problems. The machine freezes during the treatment. And the summary of this trial, first, the focus ultrasound treatment did not demonstrate significant beta seizure control in this trial in a 4-week observation. However, it seems that the seizure reduction only becomes noticeable after the 2nd month of the focus ultrasound treatment. And the effect was more pronounced when sonication targeted was in the medial temporal region compared to non-temporal region. And also the reported side effects include headaches and abdominal pain and brain fog. There was no significant change observed in anxiety, depression, and quality of life in this focus ultrasound treatment group. And this is the 3rd trial. Now we are kicking off. The first case is on the screen. We use a parallel group. One is low dose and one is high dose. The high dose and low dose is different the duration of treatments. And this is the scenario when we were using the Navifast to treat a patient with drug-resistant epilepsy. And personally, I think this field, it is very potential in this field. Why? Because it is no radiation. There is no incision and no sedation, no constraint in MRI, and a safe space. But most importantly, it's about the efficacy. And from our very early data, and also if you are very concerned about this field, Dr. Burbrick published data of 6 patients in the Brighton Women's Hospital. In her trial, all the seizure reduced and sustained for 6 months. And I think if we can enroll more data and try to seeking the best suffocation parameters, and this can help us to achieve the goal. And thank you for your listening today. Thank you.